Okay, so this is the last part of chapter 12. We'll have some review questions, and I'll tell you a little bit about telomeres and telomerase, which are important in human health and human disease. And they're actually important for all eukaryotic cells because our chromosomes are linear pieces of DNA, and linear DNA replication requires telomeres. And I'll tell you why when we get there. Okay, so let's just do a few quick questions before we get to that point. I said you need to know some of the guys in that previous segment and who did what. So let's just check and see if you recall some of this. So the first question is, who are the guys who showed that genes are made of DNA in bacteria? There's 3 of them. That was Avery, McLeod, and McCarty. Okay, who are the guys who demonstrated that DNA replication is semi-conservative? Now, Watson and Crick predicted that replication would be semi-conservative, but these guys actually showed that with density gradient centrifugation. That was Meselson and Stahl. How about the Japanese scientists who showed that when DNA is replicated, one of the strands is originally made in short little pieces that have to get tied together afterwards. And those short little pieces are named after the geneticists involved. So I gave you a hint because it's a Japanese name. Those are the Okazaki fragments. So doctors Okazaki were the ones who demonstrated that. Who are the parascientists who showed that viral genes are made of DNA? They used radioisotopes, so radioactive sulfur and phosphorus. They used a blender to do their experiment, the blender experiment. That was Hershey and Chase. Who was the guy who came up with his own rules that upon later examination, people realized that his rules implied the base pairing rules? Starts with a C. They're called Chargaff's rules. So Erwin Chargaff is the guy who came up with those. Those are the rules that the proportion of purines is always 50% of your total base content. The proportion of pyrimidines is 50% of the total base content. And the proportions of A's and T's are always equal. The proportion of G's and C's are always equal to each other. The 2 people who did X-ray crystallography that demonstrated DNA was a helix shape, although it didn't give the details of the double helix. The one was the woman who died of cancer, which probably is, it's the only justifiable reason she didn't get a Nobel Prize, along with Watson and Crick. Those Wilkins and Franklin. So Maurice Wilkins, Rosalind Franklin. And then who are the guys who are the famous ones from all of this who actually put together an atom by atom model that showed the shape of the double helix and how DNA could have a structure that fit all of the other people's observations? That was James Watson and Francis Crick. All right. I will skip over all the gossip about all of these people, but I can tell you There's just a whole soap opera of high drama surrounding the behavior of these people. Some of them behaved very, very badly during their careers, and some of them were really wonderful and heroic. But all of them did contribute very significantly to our understanding of DNA and our ability to not just understand, but work with DNA in a productive way today. Okay, so finally I wanted to talk a little bit about replication when you get to the end of a linear chromosome. And it's a problem. Replicating a linear piece of double helix is a problem because DNA polymerase can't really make it all the way to the end of the string. And yeah, so you don't get the whole DNA replicated for a linear piece. With a circle, you can do the whole thing, but linear, no. So the solution to that eukaryotic cells have is that they produce these things called telomeres, and it's a separate enzyme that builds telomeres. So a telomere is a very boring, super repetitive, like the base sequence is just really boring over and over and over again, hundreds of times, a long string of bases that get attached separate from the general process of replication. So telomerase enzyme puts on these telomeres, and they're sort of sacrificial DNA. So when a chromosome gets divided during S phase of the cell cycle, the DNA polymerase copies the most of it, but it doesn't get all the way to the end, so it doesn't get the whole telomere copied. And so every time a cell divides, the telomeres get shorter and shorter and shorter because the DNA polymerase doesn't make it all the way to the end. So after one division, it's a little shorter. The next division, even more of it's going to get lost. And after about 30 cell divisions, the telomerase is all worn away. And now, so the telomerase is fine to lose because there's nothing important in the telomere sequence. But once that's gone, if that cell continues to get replicated, you're going to be starting to lose important genes, important parts of the chromosome. And so that puts a limit on cell division so that cells can normally, eukaryotic cells can only normally divide around 40 to 60 times before they can't divide anymore because they've run out of telomeres and they're going to lose genes that the cell needs in order to survive. So how do you get the telomeres put on in the first place? Well, mostly telomeres get put on really, really early in your life during generation of eggs and sperm. Telomerase enzyme is active very, very early in your life and makes the telomeres that are going to get you through 40 to 60 cell divisions. And then that telomerase enzyme gets turned off in most of your cells for the rest of your life. And so you don't live forever. Your telomeres wear out, you get old, you die. That's called senescence. Aging and dying is senescence. Sometimes in some cells of your body, that the telomerase gene that produces that enzyme, that gene will sometimes inappropriately get turned back on later in life. And in those cells where the telomerase inappropriately got turned back on, it's going to stick new telomere sequence on the ends of the chromosomes. And what that's going to mean is that cell can keep dividing when normally it would stop. So with those the telomeres getting replaced by the telomerase enzyme, those cells can continue to grow and divide indefinitely, forever. And one of the problems with that is if you have uncontrolled, unlimited cell division, that sounds a lot like a cancer cell. So that is a problem that's commonly an issue in cancer cells, is that they have active telomerase. So it's not good to have a telomerase letting your cells keep dividing over and over and over again, because those cells then have the potential to divide continuously to the point that they develop and form a lump or a tumor. Okay, a few more little review questions. I just wanted to look at, I think I'm going to come out of my slideshow so I can just show you this and type on it. These are some problems just to let us review things like base pairing rules. So if we have this piece of DNA, these were the 2 strands of the DNA, and we were going to replicate them. Let's see if we can figure out what the new DNA would look like, and we'll make it a different color. Let's make it, the new DNA, we'll make it yellow. Okay, so remember that the 2 strands of a double helix have to be anti-parallel, so we're going to have our 3' end, oops, I'm going to make it yellow. Our 3' end is going to have to be, of the new strand, has to match up with the 5' end of the other strand. Okay, so there's our 3' end of this strand, and then the 5' end is going to be down at the end when we get there. Okay, so A pairs with T. G pairs with C. Those are the base pairing rules. That's all the base pairing rules there are. C's pair with G's. So this G is going to pair with a C. T's pair with A's. A's pair with T's. We have 2 T's. We're going to pair with 2 A's. C pairs with G. G pairs with C. C pairs with G. G pairs with C. Now we've got an A that's going to pair with a T, and then those 2 T's pair with 2 A's, and we end with a T, and this over here is the 5' end of our newly made strand. And so that strand was replicated, and then the other strand is also going to be used as a pattern or template for replication. And this time we've got the 3' end over on the left, so the new DNA is going to have its 5' end over on the left to be anti-parallel. So T pairs with A, C pairs with G. We have 2 G's. We're going to pair with 2 C's. C pairs with G. A pairs with T. T pairs with A. 2 A's pair with 2 T's. Got a T that pairs with an A. G pairs with C. C pairs with G. G pairs with C. And then we have an A pairing with the T. We've got 2 A's in our pattern or template, which is going to mean 2 T's in the new DNA. And then our 3' end is going to have an A on the end. Okay, so if we look, this was our original double helix in white. When it was pulled apart and replicated, look at what we've got. We end up with a double helix here and a double helix here. Each has the original white strand paired with a newly made yellow strand, so this was semi-conservative. Because the base pairing rules are followed when you're building the new strand with the old strand as a pattern, this DNA here has the same exact base sequence as the DNA on the bottom. So we've got 2 double helixes now, and their base sequences of the double helixes are identical to each other, and they're identical to the base sequence of the original DNA. So DNA replication will make an exact copy, or you'll end up with 2 exact copies of the original DNA, and these copies will consist of 1 original strand paired with a newly made strand. Okay, let me go back to the slideshow from here. Okay, a few more questions. What if you put a piece of DNA into a cell and that piece of DNA did not have an origin sequence? Well, I wanted to remind you, DNA cannot just be copied starting anywhere. You have to have an origin of replication for DNA polymerase to begin copying DNA. So if you put a piece of DNA into a cell and it does not have an origin as part of that DNA, the cell will not make copies of it. It'll go away pretty fast. When we do genetic engineering or recombinant DNA, one critical part of what we do is we take our gene that we want to put in the cell and we need to attach that to a piece of DNA that does contain an origin. The next question was about Okazaki fragments. On lagging strand, DNA synthesis, the DNA gets made in these little pieces of 1,000-ish bases of length, and those have to be put together to make a continuous string to make that half of the double helix. So those Okazaki fragments are tied together. Covalent phosphodiester bonds need to join those together, and those bonds are formed by the enzyme called DNA ligase, literally the tying enzyme that will tie those short polynucleotide strands together to make a single longer strand. I don't know what the question is for that. Oh yeah. No, I think I might be missing one. There should be a question here about what happens to telomeres as cells divide. And the answer is that the telomeres on the ends of chromosomes get shorter with every cell division until they're all gone. And once the telomeres are all worn off of the ends of the chromosome, the cell with those chromosomes cannot undergo any more cell division. There are cells that inappropriately have telomerase turned back on at the wrong point in the life cycle. And the chromosomes in those cells do not lose their telomeres, or they lose them, but they keep getting replaced. So those cells will keep copying their chromosomes, and the cells will keep dividing because the telomeres keep getting restored. And some of those cells with overactive telomerase will actually develop into cancer cells and form a cancerous tumor. Oh yeah, so what happens to telomeres normally is that the telomeres get shorter, but if telomerase enzyme gets turned back on, sometimes they don't get shorter, and that can give you trouble. All right, and that's the end of chapter 12. That's your introduction to what DNA is like and how it gets copied. And then the next chapter focuses on how the information in a DNA molecule can be used to allow a cell to produce proteins that it needs to produce.